Cancer has been and will continue to be one of the most challenging diseases that humans have to face. However, that's not to say we ever made any strides in the field in treating it and encountering it. In fact, we have made small steps towards in both diagnosis and in treating it. And in this particular video, we'll be going over one of them, which is monoclonal antibodies. But before explaining how monoclonal antibodies are used exactly for treating cancer, let's take a step back and look at what exactly are monoclonal antibodies. Monoclonal antibodies are identical immunoglobulins generated from a single B-cell clone. The monoclonal part simply means from a single ancestral cell, i.e. the B-cell. These antibodies recognize unique epitopes or binding sites on a single antigen. Derivation from a single B-cell clone and subsequent targeting of a single epitope is what differentiates monoclonal antibodies from polyclonal antibodies. Monoclonal antibodies also have monovalent affinity, which means that they bind to the same epitope, which in contrast, polyclonal antibodies bind to multiple epitopes and are usually made by several different plasma cells. Monoclonal antibodies used in cancer treatment are designed in a lab to target certain antigens that live on the surface of the cancer cells. By targeting those antigens, the antibodies are able to latch on to the cancer cell and act as a call to arms for other disease-fighting cells in the immune system. More than a dozen monoclonal antibodies have been approved by the Food and Drug Administration, which is the FDA in the US, to fight different types of cancer, including breast, head, neck, lung, liver, bladder, and melanoma skin cancers, as well as Hodgkin lymphoma. First tried in advanced melanoma, monoclonal antibodies have extended some patients' survival as much as 10 years. Now that is out of the way, let's explain the types of monoclonal antibodies, which are naked monoclonal antibodies and conjugated monoclonal antibodies. Naked monoclonal antibodies, or simply monoclonal antibodies, work by themselves, and they are the most common type that is used to treat cancer. One way they do this is by targeting immune system checkpoints, which are regulators of the immune system. On the other hand, we have the conjugated monoclonal antibodies. They are basically regular monoclonal antibodies that carry a radioactive substance, drug, or toxin that helps kill the cancer cells recognized by the antibody. The chemotherapy drugs attached can target the surface of cancer cells and deliver the toxic substance to that specific area. This approach can eliminate some of the side effects of chemotherapy, which can damage healthy cells when used as a single agent. Radioimmunotherapy is a treatment that uses monoclonal antibodies in combination with radiation. By attaching a monoclonal antibody to a radioactive molecule, this technique can deliver a dose of radiation therapy directly to the tumor cells. Let's explain the mechanisms of monoclonal antibody actions. Monoclonal antibodies typically use a combination of mechanisms in directing cytotoxic effects to a tumor cell. These include antibody-dependent cellular cytotoxicity, or ADCC, the complement-dependent cytotoxicity, or CDC, signal transduction changes, and delivery of cytotoxic payloads. In ADCC, monoclonal antibody bind to the antigen on the tumor cell surface, providing the target for Fc receptors on the surface of natural killer NK cells. Cross-linking of receptors triggers release of perforin and GRAN enzymes that lyses the tumor cell. Cell debris is taken up by antigen-presenting cells, which present the tumor antigens to B cells, triggering the release of more antibodies with specificities for numerous epitopes on the target antigens and cytotoxic lymphocytes, CTLs for short, that are capable of recognizing and killing cells that express the target antigen. Then in CDC, monoclonal antibodies bind to the antigen on the cell surface, which exposes binding site for the proteins that initiate the complement cascade, ultimately triggering the release of chemotactic factors and the formation of membrane attack complex with 100 angstrom pores in the cell membrane that facilitate free pressure of water and solids into and out of the cell, thus promoting target cell lysis. Then we have signal transduction changes. In this mechanism, antibodies bind to growth factor receptors, which are overexpressed in a number of malignancies. The binding of a ligand to a growth factor receptor triggers a dimerization event and activation of a signaling cascade, leading to cellular proliferation and resistance to cytotoxic agents. But monoclonal Antibody-based signaling inhibitions can occur by blocking the dimerization event or by interfering with ligand binding, which normalizes the growth rates and resensitizes cells to cytotoxic agents. Finally, there is the delivery of cytotoxic payloads, which are the conjugated monoclonal antibodies, in which they target cytotoxic agents to tumor cells. The MAB enzyme conjugate binds the tumor cell surface antigen. After unbound MAB enzyme is actively or passively cleared from circulation, the cytotoxic agent is administered in an inactive prodrug form, which is selectively bound by the MAB enzyme conjugate on the tumor cell surface, cleaving the inactivating sequence from the prodrug and releasing multiple copies of the active drug in the tumor microenvironment. So now that we've covered how they function in treating cancer, let's jump into some more recent advancements of antibodies. One major advancement is engineered antibodies. Engineered antibody is known as a modified molecule obtained by processing recombinant antibody and reassembled antibody gene via recombinant DNA, protein engineering technology, and other methods. Compared to the common antibody, engineered antibody has many advantages, such as reduce or even eliminate the human body's rejection of reaction through genetic engineering transformation, such as fully human antibodies. The molecular weight of engineered antibody is smaller, which can partially reduce the murine origin of the antibody and is more conducive to penetrating the cell wall and entering the last core part of the lesion. There are different types of engineered antibodies, including recombinant antibodies, therapeutic antibodies, and bispecific antibodies. Therapeutic antibodies are basically everything that I've been talking about for the past few minutes. They can be chimeric, humanized, or fully human antibodies characterized by much lower immunogenicity and the potential to interact more efficiently with effector cells of the immune system. Potential clinical uses of engineered antibodies in therapeutics and diagnostics varies, including immunotherapy, targeted drug or cytotoxin delivery, effector cell retargeting, and fluorescent and radio imaging. Radio-labeled antibodies are important clinical reagents for both tumor imaging and therapy and also provide an effective evaluation of pharmacokinetics, but also for viral infection, cancer, autoimmune disease, allograft rejection, asthma, stroke, and glaucoma surgery. As you can see, the applications of monoclonal antibodies are almost endless. It's just that more research needs to be done. Wow, why don't we go into some specific examples? Let's dive into lung cancer. This thing, which for the sake of this video we'll call BEZA. It is a humanized anti-VEGF monoclonal antibody developed from murine origin, which is capable of inhibiting angiogenesis by directly inhibiting the VEGF ligand. It has been proven that expression of VEGF molecules in NSCLC is associated with poor prognosis, which means that molecules have a much more direct effect on the tumor cells. This means that BEZA can be combined with drugs and help increase clinical success in the treatment. But like all types of medicines, there are side effects. In the case of BEZA, after long periods of use, it can start to cause bleeding, blood clots, bowel problems, heart disease, and even delayed healing. Overall, it is clear that the successful introduction of antibody-based protein therapeutics into the arsenal of treatments for patients has within a few decades led to intense innovation in the production and engineering of antibodies. Although there are much more for us to do, monoclonal antibodies' role in helping to treat and diagnose cancer should not be overlooked.
You are the